Okay, so now we move to the last and concluding presentation of this session, which is going to be given by Professor Joseph Kahn from Stanford University, and it's also relating to some kind of sensing. So it's for full resolution increase in scan-free single-fiber endoscopic imaging. So.
Thank you. I'd like to thank the organizers for inviting me, and I've really enjoyed all the talks today. It's a very fascinating mix of different subjects. So I'll be talking about another kind of imaging through optical fibers, which is more new than optical coherence tomography. And I'd like to acknowledge the graduate students who worked on this, and Jeff Wild, who also collaborated on this, and also funding from Corning, and more recently from Samsung has been enabling for this research. So the goal of what we're talking about is to make an endoscope that just operates with a single fiber. So you basically put a fiber, it's a multimode fiber, and you put it in a needle. And then you can imagine that you might use this for imaging in someone's brain or inside of their eye or in other parts of the body, and you could use it during surgery or for cancer detection or other kinds of studies. And let me explain the principle by which this works. So basically you have a laser source here, and you take the beam and you spatially modulate it with a spatial light modulator, and you basically launch different field patterns into the multimode fiber, and they propagate through the fiber, and they illuminate the object, and then the reflected light gets captured by the fiber and comes back, and you measure the reflected power. So you actually illuminate the object with a sequence of different intensity patterns, and for each one you measure the reflected light, and from that you reconstruct an image. So the most obvious way to do this would be to form a spot of light at the tip of the fiber and raster scan it across the object. That's the most straightforward way of doing this, and that's the way other people have done it. And we did it in a different way, and we discovered something very surprising, and that's the subject of this talk. And so let me describe some of the prior work in this field. So in the prior work, people have used fibers that support n spatial modes. Usually step index fibers are better because for a given diameter they support more modes. And not surprisingly, the resolution that they could resolve is n spatial features in the image. Because if a fiber supports n modes, then you could basically combine the modes to produce spots at different locations. They're pixels in your image. And the number of non-overlapping spots that you can form is, of course, equal to the number of modes. So they were able to resolve n pixels in their images. And there have been some very nice work published in different places. And these are just examples of some of the test images that people have recorded. So this is an optical microscope image of little plastic spheres, probably in a gelatin substrate. And then these are the images that were formed. So they had resolution of a couple meters, a couple microns. And then another example was this work that was done by Chismar and Dolakia. And they had resolution that was similar. And what I really like about their work is that they were really focused on doing the imaging fast, and they were able to image as fast as 180 milliseconds. So you could see that they're able to form a complete image in well under 1 second. Not quite real-time video, but the system could be sped up to be even faster. And another paper used a different approach. This was a team in Korea that published their work in PhysRev Letters, and they actually didn't form spots of light, but they used random speckle patterns. And the nice thing about using random speckle patterns instead of forming spots of light is that they were able to use galvanometric beam scanners to form the speckle patterns and they operate much faster than spatial light modulators. So this approach should basically be able to allow fast real-time imaging. In their actual experimental system, it took them 30 seconds to form images, but at least they demonstrated a very nice principle. And they were able to image these small features down to the micron kind of scale. So they're able to resolve a number of image features exactly equal to the number of modes that propagate in the fiber. So let me describe the experiments that we did. Now this experimental system that we have here was never intended to do fiber imaging. We built this 10 years ago to investigate principal modes and fundamental principles of modal dispersion. So when we first built this system, we were using it to transmit through long multimode fibers at very high bit rates, like 10 gigabits per second or 100 gigabits per second through several kilometers of multimode fiber. And we had this spatial light modulator here, and we used it as an adaptive optic system to compensate the modal dispersion. And then later we repurposed it to do this imaging experiment. So we launch light at 1550 nanometers, and basically it bounces off a spatial light modulator and goes into a fiber, a multimode fiber. This is a standard datacom fiber. It's actually a Corning fiber, and it's 1 meter long. And that's our endoscope. And this fiber only supports 45 modes at 1550 nanometer, 45 spatial modes. Now, first we go through a calibration stage, and in the calibration stage, what we do is we put different patterns on our spatial light modulator, and for each pattern we measure, we put a CCD camera at the output, and we measure the intensity pattern coming out of the end of the fiber. And the number of patterns that we use is M, and it needs to be at least 4 times the number of modes that propagate in the fiber, because somehow magically we're able to resolve 4 times 4n pixels in our image. Okay, we're able to record information that can't pass through the fiber. Okay, so we go through this calibration and we measure those patterns. And then in actual imaging, we put a resolution target there, and we illuminate the target by those M different patterns, and for each one we record the reflected power, and we use that to reconstruct an image. So let me describe 2 modalities of imaging that we use. The first modality of imaging is where we just formed a spot of light and scanned it back and forth. And one interesting point is that if you want to form a spot of light at a position at coordinates xi and yi, then what you want to do is form a linear combination of the modes of the fiber, the E of x and y, and each mode should be weighted by the sample of the mode pattern at the point where you want to form the spot with a complex conjugate. So that's something you learn in quantum mechanics, how to express a function in terms of a basis set. That's just an example of that. And we did not know what the modes of the fiber are. We took our spatial light modulator and we ran an adaptive algorithm over blocks in order to get it to converge to form light at a spot, with a camera being at the end of the fiber. And so we're able to form these spots of light, just as predicted. And because there are 45 modes in the fiber, we can produce 45 spots of light that don't overlap. And using this method, we can resolve around 50 pixels, depending on how you define it. 45, 50 pixels, something like that. And I want to talk about how you sample an image and how you reconstruct it, even though this is trivial in the case of spot scanning. So you illuminate the object with a series of M different patterns. M is at least 4n. And then this R of x and y is the reflectivity of the object, and I of x and y is the intensity pattern. So if you shine the intensity pattern on the object and integrate over the object, the power you collect is called pi. And so what you do is you display the m patterns, the intensity patterns, and you record the m values of the power, and you reconstruct an image. And the reconstruction algorithm is trivial. You just have pixels, and each pixel is weighted by the brightness value that you recorded. And so we call this localized reconstruction because it's so simple and local. And so that's basically similar to what other people did. But now I'm going to talk about this other method that allowed us to practically seemingly break the laws of physics, and we could record an image of information that can pass through the fiber. I mean, we didn't break the laws of physics, but it sounds cool to say that we did. So what we did is we generated random patterns at the output of the fiber. Okay? So you basically just set random phases on your SLM pixels, and you display these patterns, and that's one of them. They look like spaghetti. And then during the calibration process, you sample that pattern at L pixels, and you discretize it into a row vector of the samples. And you have m different patterns, so you end up with a matrix called I tilde, so as not to confuse it with the identity matrix. So this is the matrix of the samples of the intensity patterns. Now you're wondering why so much math, okay? Because it's all based on math. It's a computational imaging technique. Without the math, I can't explain it. Now when you sample the object in recording the image, what you do is you have the object reflectivity, and you shine each pattern on the object, and you record the reflected power that's captured by the light, and you get a single sample. And if you take m samples, you form them into a column vector called P. Okay. And then this is how we do the reconstruction. So W is going to represent our image, and if you discretize it into pixels, it's an L by 1 vector. And the reconstruction of the image follows a very simple linear optimization formula here. Okay, basically what this says is you look at all the possible images W, and you pick the image, which is W hat, the best image, such that you minimize the L2 norm of this quantity. And what that quantity is if you take a candidate image W and sample it with the intensity patterns, the reconstructed image is chosen so that you get back as closely as possible the reflected power values that you observe. Now, because of this problem is not over-constrained, you're able to find a W that gives you exactly what you observed. And you can solve this optimization problem using simple linear optimization by singular value decomposition. This is a known way to solve this problem. So basically what you do is you take the set of random intensity patterns which you've stored in a matrix, and you do a singular value decomposition of it, and then the V's are singular vectors of that matrix. You can think of them roughly as like eigenvectors of that matrix. And then the D's are the singular values, which are somewhat related to eigenvalues. And the matrix has exactly Q non-zero singular values. And though the number of singular values cannot exceed the rows or columns, whichever is smaller, but it is often much smaller than the number of rows or columns. It's the rank of that matrix, essentially. So what this expression says is basically that what you're doing here is you're taking your observed powers and you're transforming them to the basis of the singular vectors, and then you're weighting them by the inverses of the singular values. And that's exactly what that mathematically says. That's how you form a reconstructed image. So these are some imaging results. These are the worst images ever recorded through fibers in terms of, because we had a fiber with so few modes, but still we demonstrated something very surprising. So this is, these are simulations, first of all. This is some resolution bars, and this is the 50 micron core of the fiber. And if you do spot scanning back and forth in a raster, this is the image that you get. And if you use the random patterns with our reconstruction algorithm, you can resolve them clearly. So there's an obvious difference in the resolution of our technique. And then we actually did some experiments using a fiber with 45 modes, and we were able to resolve patterns that couldn't possibly be resolved with spot scanning, and we're able to resolve more pixels than the number of modes that pass through the fiber. Okay. Now, our system was very slow. It was not designed for this purpose, and it took 36 minutes to record each one of those images. I'll talk about a system that we're building now that can be dramatically faster than that. So the experimental observation is, first, we have a fiber that supports n modes per polarization. And if we use local sampling and reconstruction or spot scanning, we can resolve n pixels in the image. But using the optimization-based reconstruction, we can resolve 4n features. And obviously, we're recording information that cannot pass through the fiber. So the question is, how can you do that? It sounds like a paradox. Now, it's really not a paradox, because the number n, the number of modes in the fiber, does not limit what information you can communicate through the fiber. It just limits how much information you can send through in one shot. But if you have more observations, you can pass more information through the fiber. So the physical explanation of how this works, it's based on mode mixing. And it's really an instance of non-coherent or incoherent imaging, which gives you twice the highest spatial frequency content. So when you square a field pattern to produce an intensity pattern, you're mixing modes. Just like if you square a sine wave, you get twice the frequency. And so physically what happens during imaging is that we illuminate an object with a combination of n modes, and we can describe the complex reflectance of the object as having up to 4n modes. And so when the object is illuminated, it produces some information that's within the n modes, and it can come back through the fiber. And one of the problems in this imaging technique is that when you illuminate the object with a pattern, the fraction of light that reflects is not consistent from one pattern to another, and it tends to make the images look noisy. That's one of the things that we're working on improving. Now, there's a simpler description of this that we used in analyzing the resolution of this technique, and it's given here. Okay, so basically, imagine that you have a fiber that supports n modes, and using the SLM, you form a very large ensemble of different random field patterns, and you sample them and you do a singular value decomposition. You'll find out that the dimensionality is n. You can take 1 million field patterns and the dimensionality will never exceed the number of modes. Now, what if you look at the intensity patterns instead of the field patterns? The dimensionality will be 4n. Okay, that's because of this mode mixing. And we proved that analytically. I like problems that involve some non-trivial mathematics, and I'll show you the proof of that. So this is, in fact, the proof of that. So we're assuming this is a graded index fiber, and the modes are Gauss-Laguerre modes, and mathematically they can be expressed in terms of some Gaussian envelope and some kind of Laguerre functions here. And there's two numbers, L and M, and 2m plus L is less than some cutoff, n max. And the number of modes is proportional to n max squared, and the number of modes is capital N. Now let's say that we take a linear combination of modes and we look at its intensity, which means we take the modulus squared. And then we want to express it mathematically as a linear combination of Gauss-Laguerre functions. We can do that. It turns out that because of the squaring, the beam wave size is a factor of square root of 2 smaller. And then the number of Gauss-Laguerre functions that you need, this cutoff is twice n max, and the number of Gauss-Laguerre functions is 4 times n. So we call those the field modes, and we call these the intensity modes. They're not modes of the fiber, but they're modes to describe the intensity pattern. Okay. So they're bringing out the big guns, 1 minute left. Okay. So anyway, I'm going to just skip this slide, but I want to show you the singular values here. So if you take an ensemble of electric field patterns and sample them for our fiber, you'll have 45 non-zero singular values. And if you take an ensemble of intensity patterns and sample them, theoretically you get 153 singular values. So the squaring gives you more modes, essentially, in the intensity patterns. And these are some of the singular vectors that we observe. And the last thing I'm going to just show you is the point spread function. We're able to derive the point spread function theoretically, and we found out something very elegant, that for a graded index fiber, if you form a spot of light, the point spread function is this incoherent airy disk, the blue curve. And when you do the optimization-based reconstruction, the point spread function is a coherent airy disk, but it corresponds to a fiber with twice the numerical aperture. So what we're doing basically doubles the resolution. And so this is what we observed experimentally. The resolution goes from 0.6 lambda over NA to 0.3 lambda over NA. Now, if you know something about non-coherent imaging, you know that the transfer function is a triangle going up to the incoherent cutoff, and the point spread function is this airy disk. And then when you do deconvolution, you narrow the point spread function. And so the optimization-based reconstruction is a deconvolution, but it's done in the space of the singular vectors, not in a basis of the spatial frequencies. So what we're currently doing is basically building an experimental system that can image tens of thousands of features using green light. And then we're going to be doing experiments on biomedical imaging with collaborators at Stanford. So thank you very much for your attention.
Listen, the speaker. It's a very interesting talk to conclude the session. I will allow one short question, as we need to move to our wine reception, but also some other activity will start here very soon. So any quick question? Yes, please.
I was wondering about the calibration image that you have to take on the distal end of the fiber. Is that something you have to do constantly, or as you move the endoscope in a real application through the body and the fiber curves, does that really change the mode profiles a lot, or you have to always have that imager on the distal end?
Well, what I described was a rigid endoscope such that it won't bend, but we're working on the problem of a flexible endoscope. We're looking for good ideas if you have any.
Okay.
Okay. So I would like to thank the speaker and all the speakers of this session. And yeah, let's give an applause.